species-specific differences, individual differences. Those are the types of things that animal testing cannot account for. But every drug that's developed with the human genome in mind still has to go back to an animal test. The policies can't change as fast as the science and technology is changing. So there's certainly been a lot of discussion about how to do it. You can find 1,000 articles that have Tox 21, Toxicology in the 21st Century in their titles, National Academy reports. Virtually nothing has changed in the past decade. The big report everybody cites is the 2007 toxicity testing in 21st century. Almost nothing has changed in the decade since that came out. The problem with these approaches is that they don't talk about how to do it. They talk about what needs to be done, but it's actually figuring out how to do it that's the challenging part. It's more fun to talk about you should do all this cool stuff. The actual hard work is figuring out how to do it. So that's what this new approach we're looking into is dealing with. So this is the obvious, but it's a statement that people need to realize is that the technology is not self-implementing and the obstacles are not self-resolving. We know what the obstacles are. They've been there for the past 30 years. And it's one of those things where we just say, yeah, they're there. And they're not going to go away. We have to actually look at each one of them and figure out how to remove each obstacle. And we have to figure out how to connect these technologies to current regulatory requirements. It's not good enough just to have a really cool technology. You have to be able to tell the regulator how you're going to use it in the context that they're currently requiring testing for. Okay, so this is an analogy I like to use. If you think of the new technology as the paint can and you want to paint that old surface, that's the old technologies, you can't just put the paint on there. My son put the paint on there, but it's not gonna work. You have to prepare the surface before you can put your cool new technology on there. That's not the fun part. I mean, you need to scrape the paint off. Everybody wants to focus on the technologies, not the interfacing of actually how to make it work. So that's what this new process we're talking about is focused on, both the technology and how to actually implement it. So there are a lot of barriers that we're facing. One is institutional resistance. I mean, people don't like to change. Companies have processes in place now that they're comfortable with. Whether or not they're highly effective doesn't really matter. They're comfortable, they know the rules, they develop systems over the years to meet the specifications. They don't want to change the rules now, especially these products like a pharmaceutical that takes 10 or 15 years to get to the market. At what point do you start changing the rules? Having worked in a pharmaceutical company, I can tell you that everything is bonus-based. There's no motivation for anybody to do anything different if they know they can get their bonus if they just put the stuff over here. There's not a financial incentive a lot of times internally to try anything new that could affect your bottom line. And it goes all the way from industry to people working in the government. Regulators don't want to change the way they've done things. NIH doesn't want to change the focus of research. So all of these are things we have to figure out how to address. And harmonization it's one of those things that if you think about it, it becomes really important. We have not just EPA and FDA in the federal government. We have a lot of agencies that require this testing, and right now they're essentially harmonized in the sense that you can do one animal test and meet the needs of multiple agencies. So if you do a test for the EPA, that same test result can be used for the Department of Transportation because you're going to have to transport your pesticide. If one agency accepts an alternative and the other agency does not, it provides no motivation for a company to do an alternative test. You're going to have to do both tests. That's not financially feasible. And these are businesses that are going to do what's best for the bottom line. That's a small problem compared to international global harmonization. Even if we could get everybody in the United States to agree on accepting a test, an alternative, if Canada doesn't accept it, the EU, Brazil, China, Japan, Korea, if they don't accept it, just one of those people aren't going to do it. There's no motivation for people to have to do the animal test plus the new test. You're actually increasing the odds of failure by doubling the testing. So thinking about the financial aspects and making it easier for people to do this testing is going to be very important. We don't have human data. I think a lot of people think we do because we do clinical trials and we have some clinical studies. The amount of human data we need to actually start developing confidence in predicting human toxicity is not sufficient, not even close. There has to be an entire program focused on collecting and analyzing, storing human data. I mean, think about the challenge of that, where the federal government's going to ask for blood samples and sequencing people, and the ethical issues around that alone are mind-boggling. But it has to be, if you ever want to be human predictive, you need human data to see if your test actually works. And then my favorite is the animals as gold standard. This is one of the first major hurdles we're going to have to deal with, and we're going to have to deal with this coming year quickly. And so the principle is that you have, I'll even have a separate slide on this. So our principles for validation, this is what I call the validating alternative methods, reinforcing the misguided logic of a necessary evil, because our current validation models, policies, regulations have wording built into them that the new test has to be equivalent or better, or at least as protective of human health. 99% of these tests, we have no idea what their protection of human health actually is. You can't quantify it. A rat oral LD50 is a question of value to start with, but you certainly can't quantify how protective it is of human health. And the more problematic statement is it has to be equivalent or better. So right now we have an animal test, and we develop an alternative. Even the language alternative is reinforcing the need for animal models. We're saying that you can have this animal test, or you can have some other test that provides the same information as the animal test. All you've done there is replicate the animal results, maybe. The problem is animal tests usually can't replicate themselves very good. So we have this very fuzzy standard we're trying to hit, and it becomes impossible when you also consider that, like I said, the relevance to humans is often unknown. So consider the situation where we're in right now, where we have human data and we have animal data for the same endpoint. So skin sensitization, when you have, say, nickel, if people are allergic to nickel, earrings or rings, or latex allergies, that type of reaction is extremely important, and right now animal tests are used to do it. But it's one of the very few where we actually have human data, because we have hundreds of people that have done skin patch tests, so we can compare the two. So it turns out that the animal test, which is the standard, is about 75% predictive of the human. The animal test is also about 75% predictive of itself. So if you do the same test, you will get about 75% of the time, you'll get the same answer. To make matters worse, we have human data, we have animal data. When we develop the alternative, we have to train the models. We have to do the alternative to predict the animal results, even though we know they're not predicting the human results. That's within the legal framework of agencies. That's not negotiable. That is, it's in Europe, it's here in the United States. It just points to it doesn't make any sense. But it's a reality that we have to deal with if we want to move forward with these tests. So dealing with that is going to be a big issue. So we've actually broken up the project into two specific topics. One, or focus areas. One is what we call acute systemic toxicity. This is the kind of stuff agencies need to know if something's going to kill you quickly. So this is what you see the little placards on the side of trucks or on the side of a pesticide that tells somebody, do they need to wear gloves, do they need to wear goggles, do they need to wear a gown. this is fairly straightforward stuff compared to the more complicated endpoints like developmental toxicity and cancer. So the EPA actually requires 6 tests on every pesticide that's registered, every pesticide in every formulation. And these are listed here. And we've committed to getting rid of all of those by 2020. So if you're familiar at all with anything to do with validation, that timeline seems absurd. The simplest test will take millions of dollars in 7 years or a decade for a test that basically almost does nothing from a regulatory standpoint. So to think that we're going to replace these within, I think we can actually do it in 2 years, but I gave us 3, is pretty incredible. And the reason we can do it is because this is being driven by the agencies. Historically, all these methods have come from test method developers thinking they're going to make money by coming up with an alternative method. The agencies are put in the position of a gatekeeper. They basically are figuring out, they're already starting out reluctant and saying, trying to find reasons essentially why these products or approaches won't work, just so they can be predictive. We've had a complete flipping of that in the past couple of years. We now have agencies that really want to find alternatives, and when it starts from inside, that's the whole world difference, because by definition, whatever they think is good enough is good enough. And the Department of Defense, this is a high priority for them. They have a lot of money and a lot of interest in doing this. The EPA has publicly stated their intention to move away from these tests. So now we have 2 huge agencies with a commitment to move away from this. So they're working together to actually make it happen. But the bigger issue is how to replace all those animal tests that I showed before, that big long list. Most of those were not acute toxicity. Most of those were repeat dose, where you give the animal the same chemical over and over and over again to see what happens. That's going to take a completely different way of looking at things. It's going to be way more challenging. You notice there's no date associated with this. We're not naive enough to think this is going to happen in a decade or 20, but we have to start now identifying the key processes that are going to be required and figuring out how to work those out. So as far as this year, we are developing a white paper to send up, and I'll talk about that in just a second, but to focus on the need for doing this and identifying some key examples of why it's needed. I mentioned the reliance on animal data. We also have situations where there's no animal data and there's no human data. How do you develop confidence in a test where you don't have any records to tell you if it's toxic or not? High quality human data collecting, that's going to be a priority. And the single species safety package. So I highlighted those tests in that one slide, that same test performed on 2 species. So the theory here is if we can't at least reduce that to 1, we'll never replace animals. So let's focus on a place where we can keep one animal test in place and focus on replacing the other. Develop confidence in that. That will help us move away from both. And so the idea is to get this up to the Office of Science Technology Policy by the end of the year, so that this administration can at least get it on the docket for the next administration. And for those of you that aren't familiar with OSTP, it's the White House. It's President, Vice President. That's as high as it gets. They have the authority to tell agencies what to do, and that's really the only way this is going to happen. It starts at the very top and comes down. And the reason that the agencies are buying into this is we're giving them the opportunity to tell OSTP what they need, as opposed to just putting it up there and having the White House tell them to do it. So this is an opportunity for agencies to actually work together and tell the White House what it would take to make this happen. And it'll be interesting. You may not realize what kind of the alignment of the stars that's happened over the past 3 or 4, 8 years. So all political appointees have to resign when there's a new administration. So we have some people that have been in the same job for 8 years now. And we have a lot of very progressive people in places, and they want to make a difference. So Francis Collins would never have said that 10 years ago when he first took the job. He's leaving. He has nothing to lose. He doesn't have to follow through with that. Same with the head of the EPA. They are all in on trying to get something happening as fast as possible, and they have been over the past year and a half to 2 years. So they recognize they have an opportunity to actually make a change, and they don't have to be held accountable for it, essentially. So we need to get as much done as quickly as possible while these people are still in power, and help force the next administration to deal with some of the commitments that were made. So that's the end of my talk.
Our last panelist of the evening is Dr. Locke.
Thanks. Can everyone hear me? Okay. All right. So I want to start out by first thanking all of you for being here tonight. I know you worked a full day and then you come to hear about toxicology and ICVAN and TOSCA and FFTCA. Not the most scintillating thing. We really appreciate it. I also want to thank the organizers of this panel, Dana and Christine. Really appreciate all the work you guys did, and also inviting me up from DC. And finally, I want to thank the other panelists, because I actually learned quite a bit from them about what they do. And I know that you guys, I saw you paying attention during their talks, and I saw a couple people getting their checkbook out. So Sarah, I think you need to spell the name of your organization. And I know some of you were Googling the exciting litigation work that Anna was doing as well. So, as you heard, I'm from Johns Hopkins, Bloomberg School of Public Health. I'm an academic now, but I actually started my professional career right here in New York City. My first job was in One World Trade Center, and I practiced law there. I did financial securities law, and then I became an environmental lawyer, and then I got my doctoral degree and went into academia. So what I do now is I kind of sit on the bridge between science and law, and that's really what I'm going to talk about today. And what I'm going to do is I'm going to go over something that I think might be very exciting for us, which is this change in the Toxic Substances Control Act. Now, Anna was nice enough to leave that to me to talk about. So I'm going to try to do as good a job with this as she did with her laws. I don't know if I'm going to make it, but I want to sort of take you through this evolution of TSCA. So the Toxic Substances Control Act, or TSCA, was first passed 40 years ago. So get this image in your head, for those of you who want to think about 40 years ago. Remember John Travolta in Saturday Night Fever in that white polyester suit, right? Okay. So that's 1976, 1977. That's when TSCA was passed. And TSCA was passed because we needed to fill a gap in our arsenal of environmental laws. We had environmental laws that covered waste, we had environmental laws that covered air, we had environmental laws that covered water, but we didn't have any environmental laws that covered chemicals and commerce. So the idea behind TSCA was, hey, let's give EPA the authority to regulate these. And that's really what TSCA was supposed to do. And the idea was we could do better public health protection if EPA could regulate these chemicals. Now, TSCA was passed, and what happened when TSCA got going is the world of chemicals got divided into two pieces. I'm calling them on the slide old and new chemicals, but they were really existing and new chemicals. So what were existing chemicals? Existing chemicals were the approximately 62 to 65,000 chemicals that were used in commerce in 1976-77. And new chemicals would be any ones that were introduced after that. So the way TSCA worked was EPA created an inventory for these 60,000+ chemicals. And the inventory was out there. And if you had a chemical on the inventory, EPA their job was to go and look those chemicals over and do toxicity testing and other things, and allegedly figure out which ones were dangerous, get the bad actors off the market, and then keep the good actors on the market. So that is what the philosophy was for existing chemicals. For new chemicals, if you were a manufacturer of a new chemical, you had to go to EPA with this wonderful form called a PMN, a pre-manufacturer notice, right? and then you would show EPA that this chemical was safe, and EPA could look at what your form said, and they could say yes or no. You also had to do that if you had an old chemical where you were going to use a significant new use for that old chemical, and then of course you wouldn't put a PMN in, you'd put a SNUR in. Oh yes, I know you were thinking of that. A significant new use registration or requirement, I don't remember exactly what the acronym is, but you get the idea, okay? So that's sort of the toxicity testing piece of old TSCA. Now, were there any alternatives to animal testing in TSCA? Because obviously the idea is if you're going to have to do all this testing, 1976, 77, you're talking about animal testing. And the answer is no. There was no provision for alternatives. But, very interesting, on the TSCA floor debate in the House of Representatives, there was discussion about alternatives. So this is 40 years ago. And there were people saying, hey, maybe we should actually add provisions to the law that would force the EPA administrator to use alternatives. But after the discussion, they said, you know what, that's going to happen anyway, so we're not going to do that. We're going to give the administrator general language so the administrator can choose the best science. So that's why I put alternatives no, but, okay? Okay, so that's 40 years ago. Let's flash forward a little bit into the 80s and 90s, and let's look at what happened with original TSCA. Well, if are any of you here environmental practitioners, or have you worked with TSCA? No, you have. Okay, I'm sorry for you, but when I practiced environmental law, I thought I was lucky because I didn't have to work with TSCA. But then I get out of practice, go to academia, and I have to learn something about it. So go figure. But anyway, this is my take on kind of what sort of went wrong with TSCA over the course of the 40 years. And TSCA had a lot of problems. I can't go through all of them here, but I wanted to go through a couple. The first thing was there were no base case tests for new chemicals. So if you look at the new chemical applications that EPA got, they just have whatever information the manufacturer kind of wanted to give them. And then EPA was stuck in the situation where they had to actually go out and kind of figure out what to do with the new chemicals. Now, I took a look at that, one of my graduate students and I, and we actually found that EPA mostly used in vitro, non-animal based testing or information to make those determinations. Interesting. But there was no base case. So for new chemicals, we didn't have any standard set. The other thing was that EPA found it very hard to regulate existing chemicals. So one hope of the old TSCA was, hey, we're going to get this law out there. We're going to actually learn information about the toxics profiles of these 60+ thousand chemicals on the market. Well, that didn't work. And it didn't work for a lot of reasons about the way TSCA was structured and the regulations were written. I'm not going to go into those, and frankly, I don't really even know that I understand all of those. But there's sort of universal agreement in the legal community that it was a failure. So what you had was, as a result, over the course of these 40 years, about 5 of 62,000 chemicals were banned. And if you look at the scientific literature, probably of the tens of thousands of chemicals out there in commerce, we really only have a good toxicological profile on about 900 to 1,000, I'd say. That'd be my guess. So this created a toxics information gap. Now, when you think about that, if you want to get toxicology on 60+ thousand chemicals, and you're going to design a toxicological system to do that, do you think you're going to have one that's based on animals? No way. It would take too long, it would be too expensive, and I think as Dr. Casey pointed out, a lot of the animal studies really tell you a lot about how animals respond to chemicals, but they don't always tell you how humans respond. So we have a problem there, and I'll go back to that. The other issue with original TSCA was CBI, confidential business information. A lot of the activists in the environmental community alleged that when companies submitted data to EPA, they called all their data CBI, or confidential business information. So there wasn't transparency, and advocates in the public really could not get a lot of information on stuff that was submitted to EPA and TSCA. Finally, TSCA couldn't even, because of the way it was structured, the Fifth Circuit Court of Appeals in Texas overturned a ban on asbestos. So, I mean, gosh, if we know something about any chemical, it's asbestos, and we even have human data on asbestos. So we know what it does to you, and we know where it does it. we couldn't ban it under that. So everyone kind of agreed that TSCA needed to be fixed. So in an act of rare bipartisan agreement, TSCA was actually amended, and a law was passed and signed by the president in June of 2016. So now we have this new statute out there, and it's a major, major change. The statute itself is somewhere around 70 to 90 pages, so obviously I'm not going to go through that. But I do want to talk about some of the changes that are relevant, particularly to the use of animals in toxicology. So first, we've got this toxics information gap with old and new chemicals. So what this new law does is it says, hey, EPA, we're going to get you on a schedule, and you have to test a certain number of chemicals every year. So we're going to try to fill that toxics information gap. Now again, EPA is going to have to work out a plan to do that. That, as advocates for alternatives to animal testing, that gives us an opportunity to convince EPA that's the direction they'll go in, and they probably will have to. Also, in terms of the toxicity testing requirements, there's a new provision in this on alternatives, and I'm going to talk about that. So in this law, in fact, this is the first environmental law that ever has had an alternatives provision for toxicity or any other testing or research. So that's, well, for me, that's exciting. I hope it'll be exciting for you too. I'm going to talk a little bit about what the provision means, but before I do that, I want to show this picture. This is President Obama signing the TSCA legislation with a lot of happy legislators around him. I do want to note, just in the interest of sort of shameless advertising, that President Obama is left-handed. I'm left-handed, so I feel comfortable with that. And also, I think it's really time in the evening that I throw an absolutely useless factoid at you. So here's my useless factoid that you can take home to your families when they ask if you learned anything tonight. In the 1992 presidential election, all three candidates were left-handed. What I'm afraid is that that's the only thing you remember, but that's okay. So in the new TSCA law, there was a provision that is called the reduction of testing invertebrates. Very exciting. First of all, reduction of testing invertebrates. It doesn't cover all animals, but it covers a lot of animals. In toxicology, most of the animals that are used, I think it's fair to say, are probably rodents. Yeah, we're not using chimpanzees, at least that I know of, in toxicology. We're using smaller animals. There's also, by the way, been explosion in the use of zebrafish in toxicology. And I left some references in one of my slides, and a lot of them are academic articles, which you can ignore. But there's also a link to YouTube, which has got President Obama explaining the new TSCA law. Listen to that. It's 11 minutes, and he's really good at communicating the problems with old TSCA and new TSCA, and he makes a joke about zebrafish. So, but zebrafish are vertebrates, but that's the kind of animals we're dealing with here. It's probably rodents and zebrafish, and maybe some guinea pigs or hamsters, maybe some rabbits, probably mostly rodents. What this alternative provision says is that EPA is going to reduce or replace, to the extent practicable and scientifically justified and consistent with the policies of this title, animal tests. They're going to take existing information into account, which is very important, and it actually gets to one of the points I think that Warren made on harmonization, in the sense that we've got all this information that exists, and how can we use that information to predict what happens in the future. And also, the law requires EPA to develop an implementation plan within 2 years. So by June of 2018, EPA has to have an implementation plan to promote the development and implementation of these alternative test methods, strategies to reduce, refine, and replace the methods, and to include a list of validated alternative methods. Now, you've heard that word a couple of times, validated. I'm going to go back to that because validation and acceptance are really important. And little hint, even though a lot of this is sciencey, those are legal words. That's if you want to play in this game, that's a good place to play. So that's the provision. That's kind of what it says. So what does this mean? If you're a lawyer and you've written your check to the Chimps and you've watched the exciting litigation that Anna's doing, what do you want to do next? Okay, so how do you want to get involved in this? Because I really feel, as somebody, again, who has a legal background and a science background, that we need to have more lawyers involved in this. First thing, in the interest of full disclosure, I want to admit, this is administrative law. Is there anyone here who practices administrative law or gets involved in it? Okay. So, administrative law is different than litigation. Litigation is, death by swordplay, all right? Administrative law is death by 1,000 cuts. You go to meetings, you write policy statements, you read regulations. It's a lot of work. You really have to be there, but progress can be made. So the first thing I'll tell you is, if you get in this, please get in this for the long run. We need you to get in for the long run. And the reason that that's important is because although there's a mandate in this new statute to reduce, replace, and refine animal use, and there's a mandate to reduce the number of vertebrate animals used, it's a conditional mandate. It's conditioned on the extent practicable and scientifically justified. All right? Now, what does that mean? That means that we've got to have a list of alternative tests that are both validated and accepted. So validation is the process by which a test is deemed to be able to be predictive. As Dr. Casey said, what we want are tests that are predictive of human conditions. It's great if we can cure a mouse of a disease or a guinea pig or a zebrafish, but it don't help our species much. So we have to have tests that are actually predictive and also tests that are reliable in the sense that if one lab does them and another lab does them, they come out and that's basically the same place. Also, the tests have to be accepted by the regulatory agencies. Now, a lot of that falls in the work that Dr. Casey does, and he's didn't give himself enough credit, I don't think, because he's really turned around the ICCVAM or ICVAM, even though they have no money and no resources. They've been very effective in trying to get at these two things. But keep in mind, these are important things. These are questions that you can ask as an attorney or a non-scientist. How many validated tests do we have? EPA, FDA, have you accepted the tests as part of your regulatory arsenal? And if you haven't, why not? What's it going to take? So that's the first thing I would say. The second thing I would point out is that there is this implementation plan, and there is a process. It's going to be an open process. EPA has to consult the public. Make sure to keep your pedal on the metal there, and look at what EPA is doing, and make sure that their focus is on developing alternatives as rapidly as possible. So I think those are two things that if you want to get involved, you can certainly do. You can do that from your perch here in New York. You don't have to go to Washington, although we'd love to have you down there if you want to come. I want to take a little time to step back, though, and talk about science itself and alternatives. Because you've heard today, actually,